Bom, boa noite a todos. Como eu havia prometido no último vídeo, eu comentei que havia um professor da Case Western University que tinha resolvido fazer uma visita aqui ao Reino Unido e os senhores manifestaram interesse que eu conversasse com ele. E a conversa será em inglês. Eu resolvi gravar essa conversa ao invés de fazer um vídeo ao vivo, porque vocês podem ligar aí o tradutor quando a coisa é gravada e ao vivo ficaria um pouco mais difícil. Então, sem mais delongas, hello, Professor
Hi,
Berkley. How are you doing?
I'm great. Good to be here.
How is England treating you? Have you been enjoying your stay in this beautiful land?
Absolutely. I mean, the heat last week was a little rough, but the smell of jasmine growing on the side of the road everywhere is lovely, and the weather's now perfect. So obviously I've had a wonderful host. So thank you.
Thank you. Yeah, we had just a bunch of Lebanese food.
So we're quite full.
Yes, so yeah, we had to make an effort to be here because, in all honesty, we wanted a bed at this point in time. But here we go. A beer can keep you going. So, Berkeley, if you could just please tell us a little bit about yourself, particularly, about your Christian faith, but also what people will be very interested in, which is your line of research, what
Sure.
you've been doing over the past years, what you've been involved with.
Sure. Well, before I get started, do you mind if I say a quick prayer?
Yeah.
All right. Well, Father in heaven, I just thank you for this time, this opportunity. We look to you for insight and for wisdom, and we just ask that you would have your hand on us as we discuss all manner of topics, and we pray that it would be for your glory and the glory of your Son, Jesus Christ, in whose name we pray. Amen.
Amen.
Yeah, so to answer your question, I've been doing science ever since I can remember. I started out as a chemist, making small molecules with my own hands, designing medicines. So my undergrad was in chemistry, and then I switched to organic chemistry for my PhD because at that time I had recently become a Christian and was super fascinated by all the questions surrounding creation and evolution and like what was the science and what did it really say. So I wanted to move closer to biology, but as a chemist, there's like a wall. You can't really do biology as a chemist. And so the bridge between chemistry and biology was biochemistry, designing small molecules using chemistry that then have biological targets. So I spent 5 years designing histone deacetylase inhibitors, androgen receptor targeting agents, and nanoparticles, all manner of things. That was quite a fun time. I did that in Atlanta, Georgia, at Georgia Tech. And then after that, I went to do a postdoc at the National Institutes of Health. And that was a complete switch for me. So no more chemistry at that point. I left it behind, learned bioinformatics, genetics, sequencing. And I remember the first day I was like, what's a BAM file? I have no idea. But I was very avid and I wrote a research paper that I published during my chemistry PhD on biosemiotic entropy and the decay of genetic and epigenetic information in cancer, having no experience with it at all, but just being a fanatic of the literature and everything that was happening at the time. So I was conceptually very much ahead of the game, so I was able to hit the ground running and made huge contributions, especially in the context of understanding the epigenetic miswiring that occurs in childhood rhabdomyosarcoma. So that's a disease. It's a cancer that's essentially a muscle lineage cell that gets stuck because of mutations that prevent it from fully becoming muscle. And it's thought to arrive during key steps in puberty, which is why we only see it in a certain narrow age window. And it's deadly. It's devastating. So, we're doing everything we can to try to come up with cures for those kids and to really understand the molecular genetic basis of that disease. And when I started my own laboratory 4 years ago, we maintained that theme of trying to understand and to treat rhabdomyosarcoma, but we've branched out to a few other cancer types, osteosarcoma, which is funny enough. It's the one cancer that human children get. Dinosaurs got it. There's evidence in the fossils for that, and dogs get it quite commonly, even more common than, more commonly than children. So a few other diseases as well, but we're really excited because we've made huge advances in the understanding of the three-dimensional epigenome. So that's been the largest investment of energy and time and resources. The cell is a supercomputer, and it thinks in 3 dimensions. So the DNA is 3.2 billion base pairs long, and it folds in all these complicated ways. And how it folds determines what genes are on and off, or what gene programs are on and off, and that controls the cell's identity and fate and function.
So do you mind if I stop you right there?
Please.
So one thing that I've spoken a lot about in this channel is that this idea that the DNA is a blueprint and everything comes from the DNA is a wrong view, right? The
Sure.
DNA, for example, does not code itself. Right? I like to think of the DNA as being a database that the cell makes use of.
Yeah.
So you mentioned the 3D, shape of the DNA and how, for example, enhances sometimes are separated physically from the genes they're controlling. So, and another thing that we've been discussing in this channel quite a lot is this idea that a lot of this DNA is completely, non-functional, or what they call colloquially the junk DNA hypothesis, right?
Yeah.
So based on your research, particularly regarding non-coding aspects of the DNA, what you think about this evolutionary hypothesis that has been taken as a dogma, and in my opinion has slowed down research for quite a couple years or even decades.
Decades, yeah. Well, some background. I mean, it was in the 90s where Bill Dembski wrote saying that he was convinced because of his understanding that life was designed, that junk DNA was false, long before we had any evidence to the contrary. And that was just a philosophical implication of his view that it was designed because a designer wouldn't throw junk in. And of course, it was used by Richard Dawkins and others, mocking the creationist worldview or the intelligent design perspective because of junk DNA. They say, why would God do that? Therefore, there's no God. There was a 2010 to 2012 shift. And during that time, a couple of key technologies came online. Chip sequencing, which is the ability to sequence or to use DNA sequencing to learn where proteins are binding throughout the genome. And we had begun to understand that there's another layer of information on top of the DNA called the epigenome. So the DNA is wrapped around histone proteins. Those proteins have little Post-it notes on them that control what's going on. And so there was a summary article written in the premier journal Science that is titled "ENCODE Project Writes the Eulogy for Junk DNA." And they reported at the time at least 80% of the non-coding genome was functional. There's a couple of other really interesting things to note. Even without understanding its functions, just by doing genome-wide disease association studies, or GWAS, and just asking blindly the question, if we take 10,000 people who have diabetes and 90,000 people who don't, and just look at their genomes and say, well, where are the mutations in the diabetes patients? And this has been done for every disease, every major disease. And guess where the mutations show up?
In the regulatory regions?
in the junk DNA.
Yeah.
So 90% of disease-causing mutations are in this, non-functional DNA. Well, if it causes a disease, it's clearly functional. Furthermore, those mutations that cause diseases, autoimmune disease, I mentioned diabetes, but it's true for Alzheimer's and really the entire spectrum. Those mutations, they cluster around enhancer elements. So these are gene control elements. They're especially enriched in so-called super enhancers, which are cell type specific and often quite large and powerful enhancer elements that are off in most cells, but on and very powerful and active in the cell to which they belong. And so that's a key indicator of function. And so you have to imagine that gene control is like very, very difficult to accomplish. There's a reason why so much of the sequence of DNA is not coding. It's non-coding because control of gene expression, not just whether it's on or off, but like how much, and these control elements have to be responsive to all kinds of internal and external signals that are usually mediated through proteins that bind the DNA in a sequence-specific manner, so-called transcription factors. Well, the transcription factor code is remarkably complex, and it does require quite a bit of information, non-coding information, to accurately time the development of a baby from a single cell. I mean, the number of cascading precise events that have to occur in order for that magnificent display of unfolding biology to occur. I mean, that's all non-coding DNA in control of all those processes that look miraculous. Well, we don't fully understand it, but we know what those, we know what stages during embryogenesis. This is mostly out of studies done in mice, where they'll take a enhancer and they'll link it up to a protein that glows blue, and during embryogenesis they'll actually take mice at different stages and say, okay, when is that enhancer turning on? Where is it in the body plan? And so we have these remarkable maps now of when this junk DNA is in a very precise, both in time and in space, like in the different components of the body plan that when they sort of light up and then accomplish their goal and then quiet down. It's a very elegant and well controlled process.
Yeah, and one of these elements of junk DNA that have always been considered parasites, transposable elements and endogenous retroviruses. The more we study them, the more we realize that they're actually doing a function. They have a functional role, usually controlling gene expression. In fact, I like to remind people that the famous moth, the industrial melanin mutation,
the peppered moth,
the peppered moth, was caused by a transposable element, which I think inserted itself in a regulatory region. And the more, as I said, the more we study, seems to, because even the viruses, which is the current hypothesis for the ERVs, they are extremely important with respect to gene regulation. And it's important that people understand how your assumptions that you start with will influence how you interpret those results, right? So, for example, the fact that ERVs and transposable elements are heavily methylated and regulated by the cell was seen as an evidence that they are just, it's like the organism is trying to keep them under control because they're parasites. And the fact that under stress, this transposable elements sometimes are activated and start doing their thing.
Yeah.
And the interpretation was, well, when the cell is stressed, then it loses the ability to control these elements are damaging it.
Well, I think now there's a view that actually it's
the other way around.
Yeah, it's a very important way to buffer the cell's function. So if you have all of this transcription happening from all of these retroviral elements, that buffers the transcription of all of the other things that the cell would otherwise be doing. So it's like a brake pad almost, or like bushings. It's got like a mechanical buffering function, and because we now have these genome-wide maps of gene transcription at all different stages of life, we actually know that there are different classes of transposable elements that turn on and off at different stages of human development.
Yeah.
And so that's...
And some of them are unique to humans.
Many of them are, there's whole classes. They're called L1s. There's certain L1 classes that are very, very unique to humans. And it turns out that the ones that are unique to humans are the ones that get reactivated in certain cancers. So we have a preprint for a paper that's in review at Nature that people can look up, where we look at this in colon cancer. And it seems that they are not only turning on, but they're actually regulating other genes from a distance, which is remarkable.
But we have to be careful so that people don't think that we are just refuting ourselves and saying, well, you see, these transposable elements are actually involved with cancer. What we're saying is that they're just, like any mutation, they're just not doing what they should be doing.
Well, no, because okay. One of the features of cancer is that it would become more embryonic, more like stem cells. And so there's lots of programs that are transiently active during stages of embryogenesis and early development that get reactivated in the context of cancer. So like healthy things, good things essential for the development of life, get reactivated. Pluripotency is very important for human development. Cancer reactivates this very good and well-designed critical biological process, but just does so in a context where it's not, where those cells don't need it.
There's an error.
Yeah.
In fact, we can be a bit more like broad and say we should have probably started with it. It's like, what is cancer? Since that's the main point of your research. But before that, let me just just making a comment regarding a few things we said before. With the GWAS that you mentioned, a lot of these terms are extremely technical in our audiences. They're very just regular people who never came across a GWAS. But GWAS is just associations between genetic variants and a trait. And one thing that when you're speaking about the fact that a lot of these GWAS hits are within non-coding regions, is that another thing that is being noticed, and you know this already, is when you only look at the coding regions and you look at the GWAS peaks trying to explain the variation in the trait under the study,
sure,
it doesn't explain a lot of the variation at all. Like, so human height, apparently it's has a heritability of 80%, like 0.8, which means roughly that 80% of the variation in height could be due to genetics, at least in the population they done. I think it was a Swedish population. But when you run a GWAS, which is, if everything was about genetics and coding regions, then you would expect that all the hits would explain a great chunk of that variation. But what we find is that, in the best case scenario, you explain much less than that. And you have to look at even the whole genome, without the significant thresholds in GWAS, still explain like 40-something%. There's a lot of missing heritability that has been attributed to non-genetic elements like epigenetics and stuff like that. And with the transposable elements as well, with the stress thing, another theory that I've heard about is that it seems that the activity of transposable elements during stress is not because the cell is losing control over the transposable elements, but because it is trying to boost, they're deliberately activating those transposable elements in order to generate some variability in order to deal with stress. You see that in bacteria, for example, when they turn off the DNA repair mechanism in order to mutate more when the circumstances are extremely stressful, in order to, by pure chance, hopefully increase the probability that a mutation will show up. So yeah, going back to the basics then, because some people sent, when I mentioned you were a cancer researcher, like I remember at least one person saying, oh, they had one specific kind of cancer Hopefully the person got healed. If not, we'll pray for you for sure. Uh, so what is cancer? Like, is it one thing that just happened in different places? Are cancer different? They come in different flavors. What is the cause? Could you tell us a little bit about that?
Yeah. So every cell in our body, when it divides, it has to make a complete copy of the DNA code. 3.2 billion base pairs. It's pretty good. When it copies, it's very accurate. But there's a very small percentage of mistakes, but in terms of numbers, that could be hundreds of mutations, such that after a cell divides many times over a lifetime, those mutations, those small copying errors, they do accumulate. So the older you are, the more copying errors you have in all the cells in your body. And cancer is really just when those copying errors are in just the right places, mutating to activate certain growth genes, mutating to deactivate certain guardians of the genome that protect the cell and alert the cell to problems that are happening. Mutations that increase the number of copies for the growth genes, like MYC. Those kinds of things. When 2 or 3 or 5 of such completely random events occur during the copying errors, then that combination will give you a cell that can now all of a sudden grow without restriction. It can ignore external signals. It can often shield itself from the immune system. And so cancer is uncontrolled cell growth, essentially. And it's a consequence of bad luck, quite frankly. We know this is the case for a lot of reasons, but one, I think, interesting fact is that, almost any cell in your body can give rise to cancer. And depending on the cell of origin, you get a different kind of cancer. But not all cancers happen at equal frequency. So the kinds of cancers that we see most often in the clinic come from cells that divide much more often and make copies of themselves much faster and accumulate mutations much faster. Those are the kinds of cancers that are most common. Super rare cancers come from cells of origin that usually don't divide that much and therefore don't have that many errors or take a lot longer to accumulate mutations. So that's basically what cancer is. And so, I could talk more about it, but that's the basics.
But we are told that some mutations are magic and they just happen to, just produce some new novelty that is very good for the organism. So based on your experience with mutations, how often do they produce miracles of wonder?
Yeah, I mean, nobody no one who practices biology, medical biology for humans. So there are tens of thousands of excellent scientists who study genetics. Many of them are physician scientists, so they see patients and they treat patients. Nobody thinks of mutations as good. Everyone associates them with disease because that's what they do. Mutations are always a bad thing. There's, even the rare example of sickle cell anemia, where in that case, a mutation causes a blood cell to look like a sickle and it therefore confers resistance to malaria. Even that, you have to be like in a particular context with a particular threat to make that loss of function be accidentally beneficial, as it were. That's the only example that I'm aware of, at least in humans. But I'm aware of thousands of disease-causing, death-inducing mutations, or life-altering syndromes, that give people paralysis or blindness or all sorts of things. So
yeah, I would just make a comment. Let me just just do something here real quick. That would be better. So I hammer on this point a lot of times, and it's exactly what you just said about the sickle cell anemia mutation. People have to understand the distinction between fortuitous mutation that is actually breaking an organism, making it intrinsically worse, but that by pure chance actually help that organism reproduce and survive more given the circumstances. In the case of sickle cell anemia, you have a clear damage to the cell, right? A cell in a sickle cell format is not a good cell. It's not, you have a loss of function there pertaining to what the cell is supposed to be doing.
Yeah.
And that's the idea of teleology. Blood cells have an end, a telos, right? They're meant to do a certain thing. When that mutation happens, it is decreasing that capacity, that ability for the cell to do what it's supposed to be doing. But in the case of malaria, in specific regions of the world where, malaria is so severe, so prevalent, even though that is it's decreasing the life quality of the person, intrinsically speaking, it is still better than dying of malaria. Like malaria is a little bit worse than having the condition, right?
But we still know it's not a good thing because people in those countries, they get genetically tested before getting married because if they both have one of the sickle cell alleles and then they have a child who's got both, I mean, that's a death sentence. Like you have to have one copy of the gene functional and the other copy non-functional in order for it to be beneficial. It's not a good thing, like it's still like a scary mutation that is dangerous to accumulate in the population.
But that's the point, right? Because for evolution to work over time, you cannot just have these freak accidents that destroy the function of a thing or compromise it and just circumstantially provide some sort of a reproductive success advantage.
There's no new information there.
Yeah, you need to be able to produce something that is actually novel, right? And a lot of this this business of biologists coming to the public saying, oh no, yeah, we know this evolved in this way or this gene evolved. It's not because they empirically demonstrated any sort of mutation that led to that new gene or new cell or whatever it is, or new function. It's more a matter of just comparison, right? You have these two groups, they assume to be related. Yeah. One has a gene, the other doesn't. They would say, well, therefore, this lineage has lost the gene because the ancestor had. It's all inference that already relies on that universal common descent premise from the start, right? But you're saying that as far as you are aware, you do not know of any, let's put it this way, proper gain of function mutation that led to like a good new function. Right?
Yeah, and not only that, I think if we take a step back and we look at how many different things have to change in the code to truly get like a new body plan, like a new organ, a new capacity, like the capacity for speech, those kinds of things that discriminate, one kind of animal from another. That's a massive undertaking as far as the amount of code. And it doesn't merely involve the protein coding part. So genes that code for proteins that, they're much more, we say conserved. But
because why do we say that, right? Because they happen in many different organisms, and conservation means they were inherited and they didn't change over evolutionary time.
But if you think of it as like a systems optimization property, if you need to transcribe DNA and there's this elegantly designed, perfect fit to function protein, RNA polymerase II, that can accomplish that. It's conceivable that there's not really very many ways to biochemically achieve that goal. So if you are creating thousands of different species, and all of them have to have the same fundamental process of transcribing a gene. And the protein RNA polymerase II that is built to do that, it could be extraordinarily constrained by the desired biochemical outcome of what it's able to do, which is a remarkable thing, to be able to read the DNA and make a copy of RNA. That's I think conservation is really just design constraint. There's only so many ways to build an engine. There are physical and biochemical like rules. There's bond length, bond energy. There are torsional constraints. There's all sorts of like physics that dictate what kinds of mechanical and biochemical things need to be accomplished in order to take the DNA and to make an RNA from it. There's just not a lot of room for, like playing around with different versions. Like maybe this is the only protein that could actually accomplish this. And so therefore it's going to be pretty much the same in every species because, the biochemistry is limiting that design possibility.
Yeah, it would be a similarity due to like an analogy, right? They're doing analogous things, therefore they're similar. One challenge to what you just said that I can imagine an atheist coming up with immediately would be, you're saying that God is, if you assume these things are designed, right, and you believe the designer is a God, then are you claiming then that God is bounded by the rules of his own creation? I mean, they could say, well, why God didn't make chemistry in such a way that he could achieve the same end by multiple means, right? My answer to that would be, of course, God created a world. He could have made it in a different way, but he made it such that the world is intelligible and he has rules and patterns. And what is the problem? Why? And at the end of the day, it's a matter of people trying to impose what they think God should be doing into God, right? Oh, if I were God, I would have done it in such a way.
I hear that all the time.
Yeah, it's interesting.
That's quite an arrogant thing to say. I think that God is constrained in all sorts of ways. In the way that he has ordered the universe, like the universe has tons of constraints on it that are put in place. And I think that that's because we live in a world of order. Order is what is essential for all forms of life, and order and the information of life is essential to fighting against sort of the chaos of entropy and thermodynamic drift. So, we could talk about I think it's an interesting thing when people start to get into the theological aspects of, couldn't God have done anything because he's God? Well, maybe not. Because God himself has certain properties that are not changeable. So there's many things God cannot do. Like Jesus, for example, could not sin because he was God. God must love. He cannot choose to do otherwise. God must be just. He cannot choose arbitrarily to be otherwise. There's lots of constraints that are placed on God himself because of his character, and therefore it makes sense that if he creates a world, it will also be filled with constraints.
Yeah, I would see it a bit different. I think God is totally free, and he made the world. He could have made different worlds. He could have made a better world, in fact. That's the Catholic perspective on things. But having decided to make a world in such a way that is ordered, in fact, the fact that the world is the way it is allows for science, right? If the world was a chaotic, mass of disorder, then science would be impossible, right? Every person, atheist or not, assume intelligibility in the universe, and having done the universe in such a way, then there are constraints that are imposed by the system that God could overcome those constraints, but he would be violating the laws that he created, and that would be classified as a miracle. which is not usually how God operates, ordinarily speaking.
Well,
Like a pig, you cannot put just wings on a pig and make it fly, right? You could. God could make a pig fly with wings, but it would be a bit. I don't think the aerodynamics of a pig with wings would be very good. But anyway, let's not get too caught up with the theological aspect of this discussion.
just to get back to the point that I was getting to with the sort of it's true that proteins are very similar across species, and so what makes them unique? Well, it turns out that the dissimilarity is perhaps where you would expect it. It's in the gene control elements, the junk DNA, which of course makes sense. If you need an organism to be, functionally different, opposable thumbs versus not, or for example, the genes that control the formation of the thumb have to turn on in a different sequence, and so the information that's required is extensive and elaborate, and a lot of it's in the non-coding space.
I've seen online known biologists saying that the only reason that ENCODE, the group you mentioned before, ascribed 80% like function to 80% of the genome was because they redefined what function is, and it's too loose. There's no such thing. This thing is impossible. That is 100% functional. How would you tackle that? How do you see function, and whether there is they were just playing with the words to get the results they want?
Yeah, I mean, there's different levels of so-called function. The ENCODE project allowed something to be considered functional if it was able to, let's say, open up a small region of the genome. That's a kind of function. Or let's say there's a region that previously had no known anything, and now we know, it's transcribing this non-coding RNA. And we don't know what that non-coding RNA is or what it does, but we discovered that this chunk of the genome is transcribing that. And let's say it's transcribing it only in, certain cells, like your muscle cells at this particular step in biology. Okay, well then that's considered functional at that level. Like it's transcribing, it's doing something, it's having a function. That is a function. But what the critics would say is, that's not enough. If it's got to, if you want to define function, you can't just say, it's transcribing this non-coding RNA. You then have to go through the work of saying what that non-coding RNA is doing. How is it affecting other genes? And what they really want to see is how is it affecting the cell's function, the phenotype, and best of all, how is it affecting the function of the whole organism? So, there's different levels of proof that one is, certainly able to lob at a project like ENCODE, but ENCODE was really a cataloging project. They created all of the places of interest, let's say. And by the way, there was a follow-up called the Epigenetic Roadmap, which expanded even more on this. So they created the roadmap of interesting DNA elements that then gave the whole rest of the field of biology a catalog to kind of flip through and then chase after the function of something they found interesting. You have to understand it takes, $2 million to chase down the full phenotypic function of a single regulatory element, and a whole team of people, 6 years. But time after time after time, week after week, papers are constantly coming out saying, here's an element. We documented that it had, a low level function, meaning just it was transcribed. But we didn't know what it did. And voila, now here's this whole elaborate paper, and here's the mouse studies and the knockout experiments and blah, 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 blah. It's a lot of work. and this is the function, or at least one function that we're aware of, and boom, there's your paper. So it's going to take a long time to fully. We'll never fully catalog every function. It's not going to happen.
A lot of the papers are published, at least in my field. You don't even attempt to figure it out what those genes are doing. Like in GWAS, in humans, it's a bit easier because there's so much money being put, and it's more important for you to know exactly what those functions are, what those genes are doing. But in other no-model organisms, you're not gonna publish a paper in GWAS and then you find a few significant hits and which are close to genes, and you're gonna knock in the same project, knock every single one of those genes and figure out what they're doing. And usually those genes come from annotations from a different species, right? It's like you use a gene model. Of course, there are ways of getting empirical data for that kind of stuff with transcripts, but a lot of times you're just using, let's say in plants, you're using an Arabidopsis model, and you know that gene in Arabidopsis does X or Y, and you assume that it does the same in the different organism, which could be not the case. So people fail to understand it. It's another thing that bothers me a lot is how, in the evolutionary field, people, we know so little of even how a cell operates. And people are so quick into jumping to these huge conclusions about origins and about functionality without even understanding how those things operate. We didn't know, we knew very little about known genetic mechanisms like epigenetics and stuff up until recently.
I think that points to a historical trend that's very important to appreciate, right? That when Darwin was formulating his theory, he had no concept of genetics, really. He had no appreciation for what genetics at its core is. Very tight guardrails on what's possible for variation inside of a species. And it's governed by, sexual reproduction in most of the species that people tend to think about. And as time has progressed, our view of the cell has become increasingly complex. And increasingly sort of guarded from an information perspective. And it doesn't appear like it's going to stop. That, year after year, decade after decade, the view of the cell, especially the human cell, is mind-blowingly more and more and more and more complicated. The number of new concepts that are emerging from the study is making it difficult to actually keep up with all the functions that the cell has. So the more functionally dense and rich and complicated the supercomputer of the cell is sort of being learned about, that is making Darwin's view increasingly distant from reality, but also it is the more and more sort of engineering principles we see emerging from the study of the different control mechanisms in the cell and information relay mechanisms and decision processes that are being uncovered. The more we learn about that, and the deeper our appreciation goes, the deeper the need for this to be an extraordinarily well-designed, well-thought-out, well-engineered system. It's not, it's like the opposite of the God of the gaps, right? So in most scientific areas, it's said that the more we learn, the less we need God to explain it. So like with thunder, once we figured out a little bit of physics, we don't need to invoke Zeus or Thor to explain thunder. We can explain it with physics. And so it goes with many things, it's no longer needed to invoke a supernatural cause for things where we can understand the mechanism. But the more we learn about the cell, the greater the need is for a designer, because the quality of the design is growing year after year. And so it's the reverse. It's a science of the gaps issue. Like the more we study it, the more it demands a designer. And as I kind of stand over the precipice of like all of our understanding of how complicated the system is, I wonder, is there an end to the complexity? Is there an end to the intricacies? Or is it just infinite all the way down? Just like an infinitely complex system where we're never going to actually learn everything there is to know about it. It's
turtles all the way down.
Beauty all the way down.
Yeah. I would say only God is truly infinite, but I think we just don't have the time to figure it out, all the complexities that exist.
Yeah.
But yeah, and it's not an argument from ignorance, right? We're not saying, well, this thing, we don't know how this thing could have come about by chance and necessity, which is effectively what mutation and selection are. It's just that, it marks all those clear characteristics of intentionality, teleology. You have triangularity, you have redundancy, you have all those marks of design systems, right?
Well, it's not only that, it's information loss is happening at a much faster rate than you could imagine it being saved by information gain. So it's like, if you did not have deleterious mutations accumulating and destroying information so quickly, maybe you could imagine an extraordinary, like if we observed beneficial mutations occurring at a much higher rate than we actually observe them, by like 100 or 1,000-fold more common, and we did not see deleterious mutations, then yeah, like the gas pedal would, for evolution, would look strong enough to overcome the brake pedal of information decay. But that's just not what we observe. It's not going uphill, it's going downhill. And, the beneficials are almost non-existent. We just don't see them, but the deleterious are huge, which means it's just going, like we are observing a kind of evolution, but it's a de-evolution. Our species is losing things. It's not gaining. We can observe, and this is true in every species where it's been studied to any great depth. It's loss of information, and it's a scary thing. There are papers by Michael Lynch where he talks about the imminent extinction of the human race because we are accumulating deleterious mutations as a we're losing information as a population at a rate that's not sustainable. And so that's not something that most people are aware of, but it's out there. It's in the scientific literature. People who devote their careers to this, who are not, believers in intelligent design or any such thing, they're like, whoa, guys, this is a real problem. The human genome is unraveling. It's not going up, it's going down. So.
Yeah. I have a more optimistic view of things. I think we are the human body and biology, like there's so much complexity and so much beauty, and I think we have so many systems that are in place in order to fix those errors in multiple ways. I wouldn't challenge that things may be going on a worse direction, but I think I have more trust in the system being able to correct the mistakes. But on a positive side, when you study the cell, let's focus just on the human cell,
sure,
because that's your thing. Like, what brings you like the most? Of course, the whole thing is beautiful and it brings wonder. Like, is there anything specific that when you study it that excites you? Like, so much, and you can clearly see, that the effect of God and the grandeur of His creation. So, for example, for me, one thing that I can say is like just from the top of my head, and related to the previous topic about the mechanisms in place to fix mutations, like the ability of, even though we are talking about mutation and these errors that come, pop from time to time, the fidelity of DNA replication is astonishing, right? This thing is like a 3.2 billion. It's a book with 3.0 billion letters, and if you were to use a very good printer and print that book multiple times in a human factory, the number of mistakes would be huge, right? Because of the number of bases that you have there. But in our case, it just lacked like how many mutations every generation? A couple dozens.
Like human generations, from like parent to child, or like cell generations from parents
from a parent, like a dad and mom to the child. How many mutations?
Oh, about 300.
Okay. So,
which is very small in the bucket of the whole genome.
Yeah, billions, right? So the ability to, that precision is astonishing. We are not even remotely close to that perfection in any human system, human machine. And also the fact that the cell has those 3.2 billion letters, or 6.4 if you take both chromosomes, and they're packed into a microscopic cell, and we have trillions of cells in our body.
We have no idea. Like we can't possibly actually wrap our brains around what that means. But it sounds nice.
How long is the DNA? It's like a 1.8 meter or something, and that thing is like like folded into like trillions of cells in your body. If NCBI knew how to pack information so tightly, they would be in a good place, because how many
Bill Gates said the same thing, right? I mean, Bill Gates said decades ago that he's extremely envious of the technology of DNA. He wishes he could copy it.
Still don't. They refuse to believe that there is a source for that is transcendent. Sorry, I asked the question, gave my perspective. What is that thing that brings you the most wonder, if it's possible to distinguish all those beautiful things?
I would have to say the elegance of how the DNA is folded in 3 dimensions. It's almost like there's like a database architecture that gives the cell so a database architecture that is modular and that can change on demand, that can create quick paths to the necessary information to be retrieved for the cell's function. That is, for me, beautiful. I'm absolutely amazed by what I call mechanisms of directed motion, which we largely don't understand. But if you look inside the cell, it looks like there's a magic unguided hand that's causing the right proteins to go to the right places at the right times. Despite the fact that most things move around with Brownian random motion as their like driving force, there are some kind of unknown like zip code directions that are causing proteins to go to the right places at the right time. We now actually have some insight into some of the mechanisms by which that may be occurring. And one such exciting idea is that, how like if you take oil and you drop it in vinegar and you shake it up and you get little bubbles? Well, they're both liquids, but you have oil droplets and water droplets that don't mix. And the oil droplets, like if you put salt in there, salt won't get dissolved in the oil droplets. It'll get dissolved in the water-like vinegar or vinaigrette. And in exactly the same way, it appears that there are sort of 20 different kinds of droplets with different levels of oiliness or water-likeness, and that for each kind of droplet, they recruit by being able to properly dissolve the proteins needed for that droplet to be functional. And the best example of this is the nucleolus. So these are organelles, phases and there are different phases, and the nucleolus has actually 3 concentric phase separated. It's like a ball with a sphere around it and another sphere around it, and each compartment is separated by phase, and that creates a 3-dimensional assembly line for the processing of the ribosome and the building of it, which is absolutely amazing. And so these, it appears that biology is roughly organized by this interesting physical chemistry, biochemistry driven, not yet fully understood organizing principle.
Yeah, I was hoping that you'd say the word principle. I would say, and that's where you know already that I'm a strong supporter of an Aristotelian, Thomistic, more metaphysics, and I would say that the substantial form, the soul, is that organizing principle. Because one thing that people fail to understand is that surely you can look at the material causes that are in place within the cell, as you described. There's an oil a thing that behaves in a certain way, and they have certain properties. But the thing is that in an organism, those material properties of that substance are working for the organism. They are organized in such a way that would never happen in nature, and they are doing that for the sake of the organism. And there is a general principle that keeps all that order together for that end tellus. So, for example, morphogenesis, we know there's principles are very similar to what happens in inorganic substances, the way that the bonds are formed, and there's these waves. I forget the terminology, but it seems like the materialists would say, "Wow, the stuff that is going on at the bone level and the development level is very similar to what we see in crystals, whatever." The difference is that in the case of the bone formation development, that those physical properties are being guided towards an end that is superseding those chemical compounds and substances. So they are working for the sake of the organism, and they are transcending even their abilities, their capacities, because they are now part of a higher being.
Yeah.
That's why Aristotle says, I think physics too, that the compound, the elements work for the sake of the compound, and so to speak, right? When the elements are part of me, they take on some capacities that they would not have outside of me, because now they're working for my body. That sounds very abstract, but it's what we see.
Yeah, I think it's a helpful view because we do become, unfortunately, very reductionistic in our thinking as a consequence of the way science does move forward at kind of brute force, reductionist experiment after reductionist experiment, knocking out one gene and measuring whether or not the cancer can still metastasize to the lungs after you knock out that one gene. Like those kinds of, we're very limited in how we can study things in a well-controlled fashion.
And even knocking out a gene is not a one-to-one, right? Just because you observe an effect of knocking out a gene doesn't mean that phenotype that got changed, there's a one-to-one causality.
It's certainly not ever one-to-one, especially because the genes we like to study are transcription factors, which control hundreds of other genes. So, I mean, it's definitely never one-to-one. It's always, you perturb one thing, and then there's a whole cascade of effects, and it's very difficult to pin that down with any sort of precision. So, we are very blind, to be honest, despite claiming to have deep mechanistic insights when we write our grants and papers. But I think we could benefit as I think the scientific endeavor could benefit from some more holistic thinking.
Yeah, even a guy like I think it was Denis Noble, who is a materialist as far as I understand, even though he has some different ideas. If you read his Music of Life, at one point he's attacking. He's still a materialist, but he's not a reductionist, right? And he's describing the human physiology and the human functioning. And he reaches a point that he actually talks about there seems to be some sort of conductor, right? You have these material parts interacting, doing stuff, but there is something that is guiding those processes in such a way. And I was reading him, and I was like, oh my God, is he going to get to the substantial form or Aristotelian form and say, well, there is a organizing principle? But then, of course, he takes a complete wrong turn and start talking material nonsense again. But at least he has that understanding, right? And it's becoming so overwhelming that it's impossible to deny there is more than just the genes. And in fact, I think there was a Nature commentary that came out recently about a book that saying, yeah,
that
it's
says
time
exactly
to,
that. Yeah.
time to drop this whole gene-centric view, which is quite way too late. The fact that we're in 2025 and people are still neo-Darwinists and gene-centric, thinking, oh, it's all about genes. I mean, it's surprising how this narrative has survived for so long.
Well, it's survived for a long time because we have been able to make some substantial advances as a consequence of it. When we mapped the mutations of genes that cause blindness, for example, we found that there are certain kinds of blindness that we can really pinpoint to a single gene, and now we can use CRISPR technology to correct that gene in the eyeball of children who are affected before they turn. I think they turn blind around age like 8 or 9, fairly predictably when they have this particular mutation. And children with that, with that blindness like starting to creep up on them, who have that particular single gene mutation, we correct it, and he gets his sight back, and that's kind of a miracle. So I think, despite its flaws, and there are many, both philosophical flaws and who knows how much more advanced we'd be if we didn't take such an approach. Nevertheless, like there's been a lot of wins in the column, for gene-centric bottom-up science. So I mean, I think it's not without reason.
Yeah, no, I'm not saying that there hasn't been advances on that view, but it's a view that is quite straightforwardly false. You could take that approach because it has its methodological advantages, provided that you're dealing with a part of a whole, right? So, for example, the fact that you have cells that developed into different cells, and they have, in the human body, you have, they have the same genetic material, but they, develop to become different cells. That in itself shows that there's more to DNA than just the sequence, right?
Yeah, certainly.
And this information was always there. So we know that something's being turned on and off. Genes are being regulated in certain ways that, in a neuron compared to like a muscle cell, in which case you cannot reduce the whole explanation to the genes, because whatever is turning on and off the genes cannot be the genome itself, right? So it's stuff like that it's very frustrating that people, provided you're doing it and you know the limitations, and you just say, no, I know there's more to it. I'm just focusing on the genes because we can mathematically describe how these things work. So like, for example, population genetics, right?
Yeah.
It's very good for certain things. But the problem is when you, in the case of evolutionary biology, for example, if you're just saying, like, I'm just trying to understand how these alleles vary in a population over time to perhaps understand how climate adaptation may happen in a genome environment association study, that's fine. But as soon as you think that that's all that is, you start making claims that, well, perhaps if I keep extending this microevolution, as they call it, and this changes in allele over time, then I'm going to make a bird out of a reptile and so forth, right? And disregarding all the other complexity. So I think it's absolutely fine, and that's what they would call the methodological naturalism and stuff like that, and these distinctions. But yeah, we can, and that's an important point you made, because as soon as we start criticizing certain aspects of the scientific endeavor, people automatically assume that we are anti-science, that we're trying to obstruct scientists from doing their very good magic work, right? And people tend to forget that we are both scientists, for example. we're doing the science. I work with population genetics, for example. I see what he can do, but I also observe what he cannot do, right? And that's why I make the criticisms that I make. One thing, perhaps, for us to conclude, unless you have more interesting things to share. Or we could stay here for the whole night, but it's already 11:30. Stephen Jay Gould, in one of his books or in an interview, I don't remember exactly, he said that religion and science are two magisterium that can coexist provided they don't invade each other's spaces, which effective.
So I think magisterium is a common word for Catholics, but maybe not for me. So
Okay, let's just put it.
What is it? What's a magisterium?
Gosh, no, don't put me on the spot. I won't be able to give you a precise definition. Let's just put it this way: there are two subjects that can exist in a same individual, provided that they do not invade each other's spaces, right?
Yeah.
So effectively, what he's saying is, it's okay, it's that condescending attitude. It's okay for you to be religious, provided that when you enter the laboratory, your religiosity has to stay by the door, and then when you enter, you put your lab coat. You're suddenly a scientist. Forget about religion. So you are, a devout Christian. You don't shy away from your faith. How do you see that division?
It's totally historically ignorant to say that in the first place. I mean, I think even without a knowledge of history and the history of science, I think you could still rip that apart. I get it, right? Like, that's our culture. We're all like division of church and state kind of thing. We like to compartmentalize, but in reality, there's a tremendous power that I get and that many others have received by not compartmentalizing, but by keeping your faith at the center of a unifying worldview. Okay, so if your faith is going to be worth anything, it has to have incredible explanatory power to create a view of the world that's consistent and fits together, where there aren't any like missing pieces or things that are that you believe that are just kind of there, but they're in conflict. I think that's a miserable way to live. Not least of which because you're committing mental fallacies all the time, and I think having a religious worldview that where you've ironed out all the places where you thought, okay, maybe there's a conflict here between my views about God and creation and the way the world is, and something science has to say. If there are wrinkles, and you go through the effort of ironing them all out to where you feel like, oh, now everything is coherent, then there are two things that happen. One, you start to understand science with a lot more clarity. It gives you a tremendous focus. It also gives you a tremendous motivation, working unto the glory of God, and, is a powerful motivator. It says, I think it's in both Ephesians and Colossians where Paul says something along the lines of, don't work unto man, but unto the Lord, because from the Lord you will receive your reward. So allowing your work to be motivated by that personal relationship with God can be very powerful and sustaining, especially in something as hard as science, where failure is always around the corner, and you have to have something to sustain you through the hard times. So that's one aspect where I find it's very useful. A second aspect is worship, which means taking delight in observing and marveling at the great Creator by looking at what He has created, is a normal and healthy function of the human soul. It says in Romans chapter 1 that that creation has revealed the character of God.
Yep.
His
invisible attributes are made visible by creation. Yeah.
Yeah.
You can know the cause, or not know fully in this life, but you can get a glimpse of the cause by the effects, which is
And that's something that we need to tap into. It's very powerful. It can be very exciting. And it's just a lot of joy that comes out of that. And I think, in science, if you want to be successful, you can't rob yourself of, joy. You've got to, seek it with a whole heart. There's a couple other things too. Scientific integrity. So we're in a time now where there's such competition for grants and money that there's a huge problem with science not being reproducible. And if you are godless and you believe that there's no real accountability for anything where you don't get caught, if you don't get caught, then it's no big deal. If that's your approach to life, then, you'll take shortcuts. You may lie, you may cheat, you may hide things that you don't want to show. You could, falsify information, exaggerate, all sorts of things, bend the truth, pressure a postdoc to produce results that look a certain way, even though it may not be true, but it's got to fit this. It's got to fit the story, So there are both subtle and extremely devious ways to destroy the progress of science by not being a person of integrity. And this is a well documented, recognized pandemic in the context of science, and papers are being rescinded all the time. People are being fired because of misconduct. Scientific integrity is something that faith in Christ and the new creation that He creates in you gives you the heart that loves honesty, as God loves honesty, and that's really good for science. There's many other things too. It's like there's a huge problem of sexual harassment, and if you are filled with the Holy Spirit and you're walking with Jesus, you're going to be sexually pure. And that's very critical. That if that whole problem would disappear in an instant if everyone took their religion into their science, and actually believed it and walked it. But historically, there's a couple of interesting things, right? So, science was founded by people of faith, whether it be Sir Isaac Newton or Francis Bacon or Louis Pasteur or Michael Faraday. Tremendous minds who were very much motivated by their vision of God. Michael Faraday, who discovered electricity and magnetism, no big deal. Just the basis for all modern technology and computing.
But the atheist commentator on YouTube has done way more than that, my friend.
Oh, yeah, I'm sure
he's sharing the science, He's sharing what this guy who just mentioned, doesn't compare.
Yeah, I know. Well,
he was probably a crypto atheist who was hiding, because at the time, saying I'm an atheist would have led to persecution. You know what I mean?
Well, Michael Faraday was an elder in his church. I got a copy of a sermon that he wrote out longhand from the University of Edinburgh. Truly remarkable man, big faith. Louis Pasteur, from which we get pasteurization or pasteurized milk or Pasteur pipettes, whatever. And he's the father, and he's the father of modern microbiology. And
Mendel was the father of genetics, and was a monk.
That's right. Mendel, Gregor Mendel. That guy was impressive for what he did. But Louis Pasteur would kneel and pray at his lab bench every day. He attributed his success to his God that he worshipped in his science. So that's why I say Stephen Jay Gould's comment is it's ignorant of history, because the people who made scientific history happen absolutely were influenced by mixing their faith and their science. They brought the two together really well, and are good examples of how to live in that.
And on that note, I speak to a lot of people, particularly young people who are, some of them, they want to become scientists. They are Christians. And speaking to some of them, I realize, and not only people who want to become scientists, but speaking to people in academia right now doing science and who are Christians, they are afraid of pursuing that career, or at least expressing their faith in a more preeminent manner, because they, like it or not, even though universities were founded by Christians in the medieval times, and as you said, the men of science were guys who were in the same sentence writing about physics and God in their books.
Yeah.
Now it's undeniable that God has been pushed away from academia and from science. You cannot, you're not going to be able to publish a paper where your conclusion is, oh God, this looks so, a product of creation or something. And a lot of people fear persecution within the scientific community. They feel that if they express their faith a bit more clearly, if they were to kneel as Pasteur, was it Pasteur you said?
Louis Pasteur, yeah.
Before their desk, before starting the experiments, they would be judged for it. They may, or if they were to criticize certain aspects of the materialistic agenda and or the evolutionary theory, they would not find a position, because they would be discriminated against. So what advice do you give to these people? And also, I know you do your part as a lab PI, right? To give a safe space for people who have those views, which is an amazing thing.
Yeah,
so
I
what word
think
of encouragement would you give to this kind of students who are scared of that possibility?
I think God has a way of protecting those who He has His hand on, and my first job as a chemical engineer out of college, my boss was a believer. My PhD advisor was a believer, although I didn't know it when I joined his lab. My postdoc advisor is a believer. The chair of my department is a believer, although that's not widely known. I think the current director of the School of Medicine medical training program is a believer, and invited me to his church. There's a misconception, first of all, that believers are not in science. They're everywhere, actually. Most of them are just quiet and don't talk about their faith or their views on creation, evolution, those kinds of things. So that's first of all. It's not as lonely out there as people tend to believe, especially among medical doctors who actually, have to study human biology in all of its anatomical detail, and then have to be with patients as their parents are praying over their child who has cancer. God gets a lot more credit in the minds and hearts of medical doctors, I've noticed, as compared to, say, engineers who never meet another human. They're just coding all day long. Although I could be totally wrong. I just don't know that many engineers. But the point is that you will be surprised to find that you're not in as much of a minority as you think. That's really important to know. And you're just not going to know it unless you sort of go in with the faith that God is going to put people around you who can encourage you and strengthen you. And He certainly did that for me. And I see Him doing that for many others. And that's been a common experience. And now, not everyone has that experience to the same degree I have, but, I would say you've had a similar experience.
Yeah, I think, as we discussed a couple of days ago, I think it depends on the discipline. I think in medical doctors, as you said, they tend to be, there are more religious people there. Although one question that could be asked is, why are they being so coy about their beliefs, right? Because if you never talk about it, you could say, well, because maybe your place is not a place to talk about religion anyway. But given the oppression that Christians are going through right now, knowing that these people are out there, there are, at least in my experience, and I know in biology, the percentage of atheists is much higher than many other fields. Much, much higher, I would say.
Well, especially I would say evolutionary biology or
Yeah.
Studying natural selection, which is certainly, microevolution is a very real thing, and it's worth studying it. But you're right. I think in my field, like in cancer genetics or epigenetics, gene regulation, nobody cares. Nobody cares about Darwin. He's just not that relevant. He's just, the concepts of Darwinian evolution for cancer evolution are certainly in play, but strictly limited to microevolution. So like that is still not dealing with the origin of species. It's dealing with mechanisms of adaptation. So, at least in my field, it's just Darwin doesn't have that many fans.
Yeah.
He's just not relevant, like we're studying mechanisms.
Yeah. I think, as you said, the wonder of the human body and all the, it invites the belief that could not have come about by a random process, like an unguided process. But unfortunately, in certain fields, it is quite populated by very aggressive materialists. And that's the kind of people that I mentioned that are undermining some of the students because they do feel extremely worried about being critical publicly, particularly if they are at a lower level. Of course, if you're a lot of people are willing to share their personal views on things. If they are, after the conference, you're having a beer with them, but not in the workplace. And a lot of the students, they feel that they're isolated. And if people are not speaking out, even though they are out there, and you may, as you said, by God's grace and providence, they may bump into individuals that will help them in that path. And that, in that case, this did help happen to me. But at the same time, the persecution is real. I've experienced it. And
it can be very real. But I'll tell you that I was warned by lots of people who are prominent, big names in the creation, intelligent design space. People have written books and, been on big podcasts and all that kind of thing, who have contributed a lot. A lot of them warned me, like, keep your mouth shut. But then, after seeing me being very vocal and open, kind of just not caring what the consequences might be, some of them said, I'm really encouraged by your boldness, by your courageousness. And I was wrong, so I think we can overreact. I think it's, we've not been given a spirit of fear, but a spirit of power and of love. And I think that is a gift that we've been given. And just like one practical note, find a local church that's close to the university and get to know the people there. There will be students who are attending the same university or who are in the same PhD program, and like find them and make friendships, to stay encouraged. I just 2 weeks ago, going to my local church, which is very close, it's like a couple blocks from the university, a student who was in my lab, an undergrad, who I didn't know was a believer. I turn around and she's sitting there right next to me, worshiping Jesus. And I'm like, what? So, I think that's a just real, don't pick a church that's like really far away from your university. If you have an option to find one that's like close, where there's going to be a high population of your fellow students there, that can be a real encouragement.
Yeah. And I think, you have a point. I think, of course, we need prudence. Prudence is an important thing, is a virtue, but at the same time, we have to be careful not to transform that prudence into a vice of cowardice.
Yeah, cowardice, yeah.
And unfortunately, we see that a lot today. And I think a lot of times is a lack of faith. You're not trusting that, even if you were to lose your job or if you were persecuted, God has told us that
This is part of it. Yeah, I mean,
he was persecuted, and he said, if you follow me,
the world hates me, it's going to hate you.
Yeah. So, but it's good that you gave this positive view that, there are more people out there that can be counted on. And, like people like you, I'm pretty sure if there was a bright individual that happened to have doubts about the neo-Darwinian capacity of generating a new species, he would have a place in your lab if he was worthy in terms of his achievements, right? You wouldn't discriminate against. And that's one thing that people have to understand, is that if they run away from academia because, I don't think there's a space for me, well, how are you going to change things? Let's assume that the things is as chaotic as it is.
We need more
open-minded.
We need more extremely bright, but God-fearing people to join up. I think that would be just wonderful for many, many, many reasons. But I just wanted to quote one other passage, which is from Hebrews, where he says, "You joyfully accepted the plundering of your property because you know that you have a better and lasting possession." There's a really remarkable thing that we have access to as believers, which is the ability to respond to losing our jobs with joy. Joyfully accepted the plundering of your physical property because you know you've got imperishable property. It hasn't come to that point for most people who I know in academia who are, in fact, I don't know personally anyone who's lost their job because of their faith.
Richard Stemberg.
Well, I don't know him.
Yeah.
Right. I personally know probably 50 individuals, maybe more, who are in academia and who have strong faith, and I haven't seen any of them lose their jobs. And so, but if it did come to it, it would, I think, so what? This is, we're living in God's universe. He's in control. All things work together for good, even when your property is plundered. And I think
there's always a McDonald's close by as well for you to
That's right. You know what? You might live longer as a consequence because you won't have such high cortisol stress levels.
Yes. Yeah. Suffering for God is a good thing, and it's an honor.
It shouldn't be shied away from. Yeah.
It's an honor. Okay. Any closing thoughts? Anything that you'd like to say? Perhaps we didn't discuss. It was quite improvised. We were both extremely tired, and we just sat down and had this conversation. Anything that's missing?
Well, I would just say that, this is a very quick skim over the top. We can obviously have gone much deeper into each of these things. So if there are questions out there, just post them in the comments, and we can have follow-ups to explore in more depth any particular things that we may have glossed over too quickly. Happy to do that.
Yeah, I think it would be good to have maybe we can join in a live video at one point to have Q&A for people to clarify, or maybe you give I don't say anything and you give a talk on whatever subject you think would be interesting. Because people want to just see science being done. And as I said, having the example of good Christians in academia doing high quality research, as you most certainly do, it's very stimulating, inspiring for these youngsters who are aiming to become scientists. And as maybe I am a bit grim about things, and I tend to focus on the negative sides of things. So it's good to have someone to put me in my place with respect to certain things. But of course, people have to understand that just because Berkeley had the experience he had, for example, I know you didn't suffer persecution. As I said, I did. Depending on the department, things are different. So realities are different. But the main point that you're trying to make is, with or without it, it doesn't matter. God is in control. He's the Lord of time. He's the Lord of all things. And he will give that which will give him more glory, and ultimately will be the best for you. And if he wants you to grow in academia, he will protect you, provided you don't act like an absolute idiot and go against his will by doing things you shouldn't be doing. So it's always important to pray and consult with God. Never do anything reckless, but always do something with love. Always check whether the things you're doing is because of your own will. Are you doing it for your own ego? Are you trying to get attention? Or are you doing that because you want to glorify God in your work? in the truth, bring people away from certain errors that you think are damaging to their salvation or to their well-being. In which case, you're good to go. If you've done that, introspection, if you consulted God and you feel that you're acting prudently, then you shouldn't be afraid of speaking out. And I have many friends who unfortunately are extremely scared, and they're very bright individuals, and they could be doing a lot of good, but they're very concerned about the consequences. And I think they shouldn't be that concerned because God has already won the battle. Anyway, yeah, it was a pleasure to have you here. I hope what is the beautiful, when I told you could stay longer and you quoted that beautiful, inspiring biblical passage about hating your neighbor if you overstay.
Yeah. I don't remember the exact quote, but yeah. Don't overstay too long in your neighbor's house lest he hate you.
Yeah. So otherwise we would have multiple meetings and lives over this week. Anyway, well, thank you for giving some of your time
Absolutely,
for all
man.
the conversations we've had over the past week. I'm sure before you leave, we're going to have a couple more.
I hope so.
And let's see if we meet in the future for the listeners. Thank you very much. God bless you all.